Hi, my name is Naveen Kumar, and welcome to my review of the New York Times story, Cannibalistic Toads Reveal Evolution in Fast Motion Study Finds, and how accurate it is in respect to the article it covers, Choosy Cannibals' Targeted Consumption of Conspecific Hatchlings by Larval Cane Toads is Triggered by Species-Specific Defensive Toxins. So let's get right into it. So let's start by meeting the cane toad. It's native to South and Central America, but it was intentionally introduced to Australia as a form of pest control. It became extremely invasive because, of course, it has no natural predators in Australia. And recently, scientists have been noticing that in recent decades, cannibalism amongst the species has actually been increasing rapidly. So according to the New York Times, the article hypothesizes that the toxins called bufadienolides, which cane toads release to ward off predators, may actually trigger cannibalistic feeding behavior in tadpoles against eggs and recently hatched toads. So the method the researchers used to evaluate this hypothesis was first they tested the potential for cannibalistic tendencies in cane toad tadpoles when bufadienolides were present as opposed to when they were absent. And in the second experiment, they tested the presence of the same cannibalistic tendencies when the tadpoles were allowed access to eggs that were in the process of hatching. So the results were as follows. Experiment 1 showed that the cane toads exposed to the bufadienolides had an extremely high appetite for eggs of their own species, as well as those of native frog eggs. And that was also tested in the experiment. And experiment 2 showed that the hatching process of the eggs also elicited a cannibalistic feeding response. And that's because when cane toads hatch, they actually express bufadienolides in their own surrounding area. So the takeaways are, first of all, the hypothesis was supported by the data. Second of all, the cannibalistic response may have evolved as a response to competition induced by high population density in Australia, which is a non-native environment. And finally, this cannibalism may actually elicit the rapid evolution of defensive mechanisms by hatchlings against tadpoles that seek to eat them. So let's see how this compares to the original article. So the background is much the same. Of course, cannibalism is the result of interspecific competition, and this is observed in cane toads. So the hypothesis comes in two parts. Part 1, bufadienolide levels are expected to positively correlate with increased cannibalistic behavior against conspecific eggs and native frog eggs. And part 2 indicates that bufadienolide levels are not expected to affect the level of cannibalistic behavior against conspecific hatchlings. And that's because, again, previous literature has already showed that hatchlings emit bufadienolides when they hatch from their eggs. So 3 experiments were done to assess this. First experiment assessed tadpole predation of conspecific eggs versus native frog eggs. And we can see the controls listed here as well as the data recorded. And notice that for all 3 of these experiments, it's primarily just survival related data. So what was the survival rate, how many individuals were consumed, and how many remained at the end of the trials. Experiment 2 determined how do bufadienolide cues affect cannibalistic behavior against eggs and hatchlings. And finally, experiment 3 assessed the effect of bufadienolide presence or absence on frog egg predation by cane toad tadpoles. And again, we can see the control groups and their data recorded below. So let's take a closer look at the experiment 2 setup. So there are 3 groups. The first group is a control with no ceramic ring, and that's used to actually emit toxins if they're present. So it just contains 1 tadpole and 10 cane toad egg poles. The one in the middle is the experimental group. So it contains the ceramic ring coated in bufadienolides. And the final group is another control that contains an inert ceramic ring. So the results are as follows. Experiment 1 showed that based on data that tadpoles prefer to consume eggs of same species over native frog eggs. Experiment 2 showed that the presence of bufadienolides significantly increased the chance of eggs being cannibalized but not hatchlings. And experiment 3 showed that the predation of frog eggs increased in the presence of bufadienolides, with the likelihood of predation positively correlating to the amount of toxins present. So that's 0 milligrams versus 1 milligrams versus 2 milligrams. So let's take a closer look at experiment 2 through this figure. So you'll notice that the predation rates when the toxins are present are much higher than those when they're absent until they hatch. After that, it achieves almost total parity. And that's because, again, bufadienolides are released during the hatching process, so it basically negates the initial absence of the toxins. So there are several takeaways from this. So we can see here that the hypothesis was obviously supported by the data, and that applies to both points. And also statistical analyses from experiment 2 that showed the presence of toxins amplifies the cannibalism of eggs, but not the cannibalism of hatchlings for reasons we've already discussed. And finally, one possible source of error is that the relatively low survival rate of the conspecific eggs compared to native frog eggs in experiment 1 is because cane toad eggs hatch earlier, which means there's more time for them to be consumed. So is this story accurate? Let's discuss accurate points first. So first of all, the article in the New York Times discussed the paper background and discussion of evolutionary advantages very clearly. It also correctly described bufadienolides and their role in the hatching process, meaning that they're emitted when the hatching process occurs. And the main takeaway of the results was stated properly, which is that the hypothesis was supported by the data. And keep in mind that they also accurately discussed the hypothesis. So there are some inaccuracies. First of all, they made no mention of experiment 3, which is a major omission. Furthermore, they also omitted major parts of experiment 2, which is specifically the presence of these ceramic rings and those controlling experimental groups. And finally, the second experiment, as described by the New York Times article, is more of a result from experiment 2 than an actual experiment. However, we have to keep in mind that at the end of the day, New York Times is directed towards a lay audience. So because it captured the overarching points of the article well, we can say that it is mostly accurate. Thank you for watching this video. And here are my credits and acknowledgements. This video was produced by Bio 182 at Arizona State University in the spring 2022 semester.